Hello everyone, I am SARITA RAWAT, Assistant Professor, School of Pharmacy, Suresh Gyan Vihar University, Jaipur, Rajasthan. Today we will start our next lecture of Pharmaceutical Microbiology. Today's topic is Animal Cell Culture Techniques. So, learning out objective of today's lecture is to study about animal tissue culture, to study animal tissue culture advantage and disadvantage, to study about the techniques of animal tissue culture. Learning of outcomes of today's lecture is you are able to know about the animal tissue culture, advantage, disadvantage, techniques of animal tissue culture. Now coming to the introductory part, so the cell culture can be defined as the process of cultivating cell and tissues outside the body of an organism that we also call in vitro studies and in an artificial environment which stimulate the in vivo conditions such as temperature, nutrition and protections from the microorganism. So we can say in cell culture we will cultivate the tissue outside the body, that is known as in vitro studies, by providing them the artificial environment as similar as the in vitro conditions such as maintaining the temperature, providing the particular nutritions and protections from the microorganism. So the cell culture was first successfully undertaken by Rosen Harrison in 1907 and the Rolex in 1885 for the first time maintained embryonic chick cells in a cell culture. Now, talking about the historical events in the development of cell culture, so it is started from the 1878. The Claude Bernard proposed that the physiological system of an organism can be maintained in a living system after the death of an organism. And in 1897, Loeb demonstrated the survival of a cell that is isolated from the blood and connective tissue in the serum and plasma. In 1907, Harrison cultivated frog nerve cells in a lymph clot that had by the hanging drop method, and he observed the growth of the nerve fiber in vitro by several weeks, and he was considered by the he was then considered by some as the father of cell culture. Now, in 1960, Ross and Jones introduced proteolytic enzyme trypsins for the subculture of adherent cells. In 1940s, the use of antibiotic penicillin and streptomycin in the culture medium decreased the problem of contamination in cell culture. In 1952, George Cryo established a continuous cell line from a human survival carcinoma that is known as Helen-Ley cells. So he established the line from a human cervical carcinoma, which is known by the HeLa cells. Then Joel Becko developed the plague assay for the animal virus using conflict monolayers of the cultivated cell in the same year 1952. In 1955, Eagle studied the nutrient requirements of selected cells in culture and established the first widely used chemically defined medium. In 1961, Haflix and Morad isolate the human fibroblast WI38 and showed that they have a finite lifespan in culture. So these are some historical aspects of the historical events and development of the cell culture. Now, the major developments in the cell culture technology is the first development was the use of antibiotic which inhibits the growth of contaminants. The second was the use of trypsins to remove the adherent cells to subculture further from the culture vessels. And the third was the use of chemically defined culture medium. Now, talking about some terminologies that come into the cell culture is the first one is primary cell culture. So, the primary cell culture is when cells are surgically removed from an organism and placed into a suitable culture environment, they will attach, divide, and grow there. Then the second term is cell line. The cell line is when the primary culture is subcultured and they show an ability to continuously propagate. Then the third term is ancogate dependency. So ancogate dependency means cells that grow as monolayers adhering to the substrate, that is glass and plastic. Then another term is passing and subculturing. So passaging is the process of splitting the cells. Then the finite cells. So finite cells mean when the cell has finite lifespan. Then the another term is continuous cell lines. So the continuous cell line is when the cell can grow into the infinite lifespan. Now, as you can see, this is the tissue culture laboratory design and it includes the cold room, the storage, fertilizers, nitrogen, freezers, air exit, and then it has sterilized glasswares, solid items, sterilized liquid, sterilized plastic, and cylinders. Then it has service chart, incubators, cell counter, aseptic area, laminar flow hoods, incubators, sterilized glassware and plastic over the bench, and this is the waste bench, and it has also the hot room, microscopic room, and quarantine room. Then it has the storage. It has storage and preparation areas, water purification areas, and corridor, ovens and autoclaves. Here is the air exit, and this is wash up areas, dryer, glassware washing machines, and shrinks. So this is the whole layout of the tissue culture laboratory. Now the equipment that are used in the tissue culture laboratory are laminar cabinet, that is the vertical LAF, are preferable and they are fitted with the HEPA filter and UV filters. Incubation facilities, the temperature of 37 degree, CO2 2 to 5%, and 95% air at 99% relatively humidity. So as you can see, this is the equipment of laminar cabinet. Now the another thing is refrigerators. So the refrigerator is used to keep the liquid media at 4 degrees Celsius and enzymes like trypsins and the media component, for example, glutamate, and serum at minus 20 degrees Celsius. Then the microscope and at an inverted microscope with 10x to the 100x magnifications is required. Then tissue culture wire, culture plastic wire treated by the polystyrene is used. So as you can see in this picture, now these are some another equipments that are used in the tissue culture laboratory. So this is the centrifuge machine, the hot air oven, and then we have lots of another machines. Then the tissue culture media. So cell have complex nutritional requirement that must be met to permit their propagation in vitro. Previously, scientists employed the chick embryo extract, plasma, sera, and lymph, etc. However, they varied into their growth promoting characteristics and thus hampering the reproducibility of the experiment. And today, a number of chemically defined formulations have been developed that support the growth of a variety of established cell lines. As you can see, this is the egg, and this is the final cultured embryo. Now, Eagle Basil's media, Eagle's medium essentials media, Dubaco's modified essential media, and Scope's modified Dubaco media, and Roosevelt Park Memorial Institute and HAM F12. The various nutrition required are glucose, fats and fatty acids, lipids, phospholipids, and sulfolipids are required, ATP and amino acid are required, vitamins are required, and minerals are required. Then when we talk about the serum, so serum can provide the various growth factors like hormones, cell adhesion factor, and other factor needed by the most mammalian cells for their long-term growth and metabolism. So FCS and FBS, CS, HS, HOS required. Now, when we talk about the properties and special requirement for media are pH. So pH should be optimum pH between 7.2 to 7.4 is generally needed for the mammalian cells, and phenol red is used as an initial indicator. So as you can see, in pH 7.8, the color of the media will be shown as pink and purple. Now, talking about the oxygen, so oxygen, the cell depends upon glycolysis for the supply of oxygen. Selenium controls O2 diffusions, and glutathione act as the free radical scavengers. And the temperature, when we talk about the temperature, so the optimum temperature of mammal is 37 degrees Celsius, and the change of plus minus 5 degrees Celsius is acceptable. Then the humidity, so for the cell growth, 100% humidity is essential to reduce the evaporation of the media, and antibiotics require penicillin 100 μ per ml for bacteria, streptomycin 100 mg per ml for bacteria, gentamicin 50 mg per ml for bacteria, and niacin 50 mg per ml for fungi and yeast. Now the limitation of serum, so it is undefined with the respect to its chemical compositions. It can be a source of adventurous agent and thereby products, and serum also presents a variable performance of cell growth and a subsequential cost. The problem during downstream processing and availability. So these are the limitation of the serum. Now, the serum-free media are the important amino acid and some trace element, growth factor, hormones, transport protein, and adhesion factor are added. Adhesion factor added are main component of intracellular substance and the serum such as fibronectin, collagen, and laminin. Primary purpose of introducing SFM is to promote the specific growth of particular type of cells. Then the type of tissue culture we have is the cell culture. So it is a tissue from a explant in the dispersed mostly enzymatically into a clinical suspension which may then be cultured as a monolayer or suspension culture. So the advantage of cell culture included development of a cell line over several generations. Scale up is possible, absolute control of physical environment, homogeneity of sample, less compound needed than in the animal model, and disadvantage of tissue culture included the cell may lose some definited characteristics and hard to maintain, only grow in the small amount of tissue at high cost. It is a de-differentiations and instability annulopity is there. Then when we talk about the explant culture, so explant culture in the growth and of the tissue of cell separate from the organism. This is typically facilitated by a use of a liquid, semi-solid or solid growth medium such as broth of agar. Then advantage of explant culture included some normal functions may be maintained better than the organ culture for scale up but not ideal. Disadvantage included original organization of the tissue is lost. Now, when we talk about the original culture, organ culture, so organ culture, the entire embryo or organs are excised from the body and culture. Advantage is normal physiological functions are maintained in this culture. Cells remain fully differentiated, and the disadvantage of organ culture included it has scale up in not recommended. Growth is very slow. Fresh explanation is required from every experiment. So these are the difference between the three cultures: cell culture, explant culture, and organ culture. So every culture has their advantage and disadvantage included. Now, as you can see, this is a culture media. So we have taken a embryo or a adult or a egg. So these are the three kind of culture, and then we dissection them. Then the finally chopped and enzyme digestion we follow. Then the further digestions if necessary, and then the cell culture present. Then the primary explants and organ tissue organ culture is required. So as you can see the difference and the results. Now coming to the primary culture, when cells are surgically removed from an organism and placed into a suitable culture environment, they will attach, divide and grow. Most of the primary culture cells have a finite lifespan of 50 to 60 diversions in vitro, and Primary cells are considered by many researchers to be more physiologically similar to in vivo cells. Due to their limited lifespan, one cannot do long-term experiment with these cells. Now, as you can see, the tissue explants are exercised using scalpel, and the mechanical disruptions by the pestle and mortar then filtered using 0.22 mu filtered to a syringe. Then enzymatic digestions by trypsin or collagenase. Cells are counted on a hemocytometer 1 to 2 into 10 to the power 5 cells per ml seeded into the media. Then 5 ml of cell is suspended into 25 centimeter into 2 flasks, and the flasks are incubated in a CO2 incubators. The flasks are observed daily for their normal growth characteristics, and media is changed every 2 to 3 days until the cell attain 80% of confluent. Now, this is the isolation of tissue. So, first step for the isolation of tissue is followed: dissection or selection of required tissue, removal of fatty acid and necrotic cells. Then, fine dissection. So, it is done by chopping down the explant size, primary explant, then explant and transfer, then secondary explant culture, and in explant also the outgrowth, and it go to the subculture and then cell line made. Then when we talk about the mechanical dissection, so it is by saving syringe or the vigorous planting, so then it goes to the dispersing primary culture. Then when we talk about the enzymatic disaggregations, so it involved cold trypsins, warm trypsins, collagenase. So when we talk about cold trypsin, so it will take overnight cold short incubations, then resuspend and seeded. And then when we talk about the warm trypsins and collagenase, so warm trypsin take long incubations, respects mating, and collagenase take long incubations, complete medium, which is then centrifuged and then sent to the resuspended and shedded. Then it goes to the dispersed primary culture, then subculture, and then cell lining. So this is whole process of making the cell lining. Starts from the isolation of tissue and dissection. Now the continuous cell lining. So most cell line grow for a limited number of generations, after which they cases comprehension of normal and transformed cells. So normal cells are encollars depend expert blood cells. So they are molecule, finite number of divisions, content inhibitions, monolayer culture, dependent on the external growth factor, signals for the proliferations, greater relations of differentiated cellular functions, display typical cell culture, cell surface receptor. In transformed non-oncologic dependent, for example, suspension and culture possible, and they are important or continuous cell lines, no content inhibitions and multi-layer culture required. may not needed an external source of growth factor, and typically loss of differentiated culture functions and cell surface receptor display may be altered by this. Now, these are some common cell lines. So, these are the human cell lines. In leukemia, you can see, and then the HeLa epithelial, then BAE1 endothelial, then MCF7 breast, and this is the HT1080 kidney cell lines. This is the fibroblast cell line. This is the neuronal cell line, and this is the lung cell line. And the cost 7 African green monkey kidney cells, and the others such as CHO from the hamster and SF9 and the SF21 from the insect cells. Then when we talk about the subculturing, so we have to remove spanned media from the culture vessels, then add the pre-warmed dissociations reagents such as trypsins, and gently rock the container to get complete coverage of the cell liner. Incubate the culture vessels at room temperature for approximately 2 minutes. Then add equivalent of 2 volume of pre-warmed complete growth medium. Disperse the medium by pipetting over the cell layer surface at several times. Then split the cell into 2 or 3 flasks containing complete media and incubate the cells. Then cryopreservation method. So in cryopreservation, remove the growth medium, wash the cell by PBS and remove the PBS by the aspirations. Then dislocate the cell by trypsin, dissociate the cell by trypsin, dilute the cell with growth medium, centrifuge at 200 gram for 5 minutes at RT and remove the growth medium by aspirations. Then resuspend the cell in 1 to 2 ml of freezing medium containing DMSO. Transfer the cell of cryovessel and incubate the cryovessels at 80 degrees Celsius overnight, and next day transfer the cryovessel to liquid nitrogen. Then detections of contaminants. So in general, indicator of contaminations are turbid culture media, change in the growth rate, abnormally high pH, poor achievement, multinucleate cells, and then graining cellular appearance, vascularizations, inoculations, bodies, and cell lysis. Yeast, bacteria, and fungi usually show the visible effect on the culture changes in the medium turbidity or pH. Mycoplasma detected by the direct DNA staining, which indicating interslating the fluorescence substance, for example, halocest 33258. Then mycoplasma also detected by the enzyme immunoassay by specific anticia or monoclonal apps by PCR amplifications of the myoplasmal RNA. Then the best and the oldest way to eliminate contamination is to discard the infected cell lines directly. When we talk about the in vitro transformation of cells, so it is transformed initiate of the established cell and changed from the normal cell to cell with the many of the properties of cancer cells. Some of these cell lines have actually been divided from the tumors or are transformed spontaneously in culture media by mutations. Chemical or gamma rays treated cell can become infinite with the loss of growth factor. Viral infections with SV40T antigen can inert oncogenes and lead to gene alterations. No matter how transformations occur, the rule is a cell with the altered functional, monological, and growth characteristics. The advantage of tissue culture include control of the environment, characterizations and homogeneity of the sample, economy scale of mechanizations, in vitro modeling of in vivo conditions. The limitation of culture media included to grow cell outside their normal environment. Three major controls are involved. The first one is observing strict abscess, providing the right kind of physico-chemical environment, nutrients in its simplest observable form. Culturing techniques needed a great deal of expertise. Tissue sample consists of a mixture of heterogeneous cell populations. Continuously growing cell often show genetic instability, difference in the behavior of cell in cultured and in its natural form, and should include the proper balance of the hormones. Now, talking about the application of cell culture, so application of cell culture include excellent model system for studying. So the normal physiology, cell biology and biochemistry of the cell, the effect of the drug, radiation and toxic compound on the cell, study mutagenicity and carcinogenicity, used for the gene transfer studies, large-scale manufacturing of biological compound and vaccines, insulins, interferon, and the therapeutic proteins. So they are used for the manufacturing of vaccines, insulin, interferons, and other therapeutic proteins. So as you can see, this is the product and this is the use of the cell culture. So as you can see, this is the monoclonal antibodies, then hormones, immunobiological regulators and virus vaccines. So, large molecule 50 to 200 amino acids produced by the hormone synthesizing organ may also produced by the chemical synthesis, for example, erythropoietin. Then it produced by the hybridoma cells and used for the diagnostic assay systems, and that is determine the drug, toxin and vitamins, therapeutic process purpose and biological separations, chemographic separations to purify the protein molecules. Also it is used as the prophylactics of the virus is collected and inactivated and used as a vaccine. A weakened form will induce a protective response but no disease. And interferon anti-cancer glycoprotein selected animal cell or recommended bacteria. Lymphokinins are interleukins that are used as an anti-cancer agent. Then the embryo and the product. So the urokinase, rannins, asparaginase, collagenase, pepsin and trypsin, etc. are used. Then the production of some insect virus, they are highly specific and safe to the environment. Also the insecticides and the whole cell and the tissue culture include artificial organs and semi-synthetic bone and dental structure are prepared. So this is all about your tissue culture, animal cell culture. Now we have some questions including that are related to this lecture. So you are while going through the lecture, you are easily able to answer these questions. So what media is used for animal cell culture? What are the type of animal cell culture? What is animal cell culture? What is a cell line? What is L-glutamine used in cell culture? In which phase of growth is the specific growth rate of the animal cell culture? How can we preserve animal cell culture? How do cell culture grow? Why do we freeze cell? Why is it important to freeze the animal cell? Now, talking about the reference for today's lecture, so if you go through if you want some other knowledge or deep in-depth knowledge about the cell culture or animal tissue culture, their types and the techniques that are used, so you can follow these references, which included Bacchus, D. Song, L. Tuna, RS 2004, adult mesenchymal stem cells, characterizations, differentiations, and application in the cell and gene therapy, which is in the Journal of Cellular and Molecular Medicines. Then you can follow the Buchler M in 2005 animal cell culture, recent achievement and perceptives in the production of biopharmaceuticals, that is in the applied microbiology and biotechnology. Then you can follow the Franke culture of the animal cell in Hoboken. Then you can follow Ganglel S, that in the principle of practice of animal tissue culture and universities. And you can also follow the biotechnology applications animal cell culture. Then Mortensen's BZ, you can follow polio and rabies vaccines produced in the contagious cell lines. And then integration of cell culture and microfabrications technologies, you can follow the provides a new manufacturing system for the production of influenza vaccines, designing cell compatible hydrozoles for the biomolecular applications, recommended protein therapy in the cultivated mammalian cell in nature's biotechnology. So for the deep knowledge or for the more knowledge, you can definitely follow these articles, these references, and we will go through just a quick review what we have studied today, so you can recall all the things and you can answer the previous quiz questions very easily. So today we have studied about the cell culture. So we have started from the introductory part. So cell culture can be defined as the process of cultivating cells and tissues outside the body of an organism in vitro, and is an artificial environment which stimulate the in vivo conditions such as temperature, nutrition, protections from the microorganism. And cell culture was successfully undertaken by the Ross Harrisons in 1907, and Rocks in 1885 for the first time maintained the embryonic cells in a cell culture. Then we have learned about the historical developments of the cell culture. So in 1878 and in 1897, then in 1907, Harrison cultivated frog nerve cells in a lymph clot that is held by the hanging drop method and observed the growth of the nerve fiber into in vitro for several weeks, and he was considered by some as the father of cell culture. And in 1916, Rose and Zons introduced proteolytic enzyme trypsins for the subculture of adherent cells. In 1940s, the use of the antibiotic penicillin and streptomycin in the culture media decreased the problem of contamination in the cell culture. In 1952, George Gray established a continuous cell line from a human cervical carcinoma known as HELA, Helene-Lai cells. Then in 1961, Hayflick and Morad isolate the human fibrosis. Then what are the major developments in the cell culture technology? So the first development was the use of antibiotics, which inhibit the growth of contamination. Then the second was the use of trypsins and remove of adherent cells to subculture the further from the culture vessels. And the third was use of chemically defined culture medium. Then the terminologies that are used in the animal cell culture are primary cell culture, cell line. So cell lines are basically the when the primary culture is subcultured and they show an ability to continuously propagate. Then anchorage dependency, passive subculturing, finite cells, continuous cell lines. So this is whole chart, whole library designs of the tissue culture laboratory. So you can see it include the cold room, storage, and then hot room, microscopic room, and quarantine room. Now these are the equipments that are used in the culture laboratory. So laminar cabinet are used with the vertical LEF are preferable and they are fitted with the HEPA filters and UV, and the incubation facilities at the temperature of 37 degree, and CO2 is available at 2 to 5% and 95% air at 99 relative humidity. Then all refrigerators are all also there, microscope is also there, then tissue culture wire is also there. So these are some instruments that are used in the laboratory, tissue culture laboratories. So what is tissue culture media? So basically the cell have complex nutritional requirement that must be met to permit their propagation in vitro. And previously scientists embryed chick embryo extract plasma, sera, lymph, etc. However, they varied in their growth promoting characteristics and thus hampering the reproducibility of the experiment. And today a number of chemically defined formulation has been developed and support for the growth and variety of the establishment of cell linings. So this is whole process about the embryo making from the egg. Now the requirements, special requirement for preparing the media. So the pH, the optimum pH between 7.2 to 7.4. Then oxygen is required, temperature is required, the humidity is required, and antibiotics are required. So for penicillin, 100 unit per ml for bacteria, streptomycin 100 mg per ml for bacteria, gentamicin 50 mg per ml for bacteria, and nystatin 50 mg per ml for fungi and yeast. Then limitation of serum. So limitation of serum included, it is undefined with the respect to its chemical composition, and it has been a source of adventurous agent and thereby the product. And the serum also present a variable performance of cell growth and at a sustainable cost, problem during downstream processing and availability. So we need a serum free media. So serum free media is important amino acid and some trace element, growth factor, hormones, transport protein, and adhesion factor are added in that. Adhesion factor added are main component of the intracellular substance and serum such as fibrinogens, collagen, and lamination. So then we have studied about the type of tissue culture. We have the cell culture, then excipient culture and organ culture. Then we have also read about its advantage and disadvantage. And then we have studied about the primary culture. So when cells are surgically removed from an organ and are placed into a suitable cultured environment, so they will attach, divide, and grow. So this is the whole process behind that. Then the continuous cell lines, normal and transformed, we have studied. Then common cell lines, we have human cell lines like leukemia, endothelial, breast, kidney, and neuronal and lungs. Then this is the subculturing process and detection of contaminant, in vitro transformation of the cell, advantage of tissue control, tissue culture, so it controls of the environment, characterizations of homogeneity of the sample, economy, scale and mechanizations, in vitro modeling and in vivo conditions. Then some limitations are also there. So to grow cells outside their normal environment, their major control are involved, like observing strict asepsis and then nutrition in its simplest observable form. Cultivating technique needed. Tissue samples consist of a mixture of heterogeneous cell population, and it should include proper balance of the hormones. So they have excellent model system for studies and use for genetic studies also. So this is all about your animal tissue culture. Thank you for today's lecture. Thank you for kindly listening to me.